Hello and welcome to Nikolay's genetics lessons, and today we have another case study, and here's a question that I got from one of my subscribers. Is it possible to get different size PCR amplicons from the same primer and DNA template? And here's a question. I'm working on OMI set strain on two sets of different primers, one in ribosomal DNA and another one on mitochondrial DNA. I also did DNA serial dilution, so the sample concentrations are different, but they are originally from the same tube of DNA template, and here you see different concentrations. After I checked the PCR result by gel electrophoresis, I noticed the different size of fragments with the same set of primer and same DNA template, but different dilution. What might be the reason? And here is the gel which this researcher refers to. So yes, it is possible to get different size of PCR amplicons from the same primer and DNA template if the PCR conditions are not optimized or there are genetic variations within the DNA template. PCR relies on specificity of primers to anneal to the target DNA sequence, and efficiency of the amplification process is influenced by the factors such as primer concentration, annealing temperature, extension time, and the quality and quantity of the template DNA. Subtle variations in these parameters can affect the specificity and efficiency of the amplification, leading to the generation of different size of the PCR products. And don't forget that DNA template itself can be heterogeneous, containing polymorphism, deletions, insertions, or rearrangements that affect the size and structure of the amplified product. These genetic variations can arise from natural variation, mutation, or DNA damage, and can be present in different samples or even different regions of the same DNA molecule. Therefore, it is important to optimize PCR conditions and validate specificity and reproducibility of the amplification to ensure the reliable results. So you can obtain such results when you use different concentrations, and also don't forget that sometimes those bands that is closer to the edges of the gel would move slower than those in the middle. So it's quite usual that highly concentrated samples migrate in the different pattern. Sometimes our machines are not as precise as they should be, and the only way to really confirm you have the same product and make sure that there is no contamination in the products, you have to do sequencing. I would also recommend to try different agarose concentration gels and see if they keep having different migrations. So this would be my recommendations, and also I would increase annealing temperature during PCR to avoid non-specific annealing of primers. You also might try such technique as touchdown PCR, which also should provide you better results. And this is all for today. Subscribe and see you in the next video. Goodbye.